Students, in this video we are going to discuss few points regarding the DNA fingerprinting. This video is the continuation of the last session. In the last video we have discussed some of the points in the DNA fingerprinting, and this video is continuation of the last video. So DNA fingerprinting, this technique was first developed by Alec John Jeffries. So Alec John Jeffries first invented this technique. That's why Alec John Jeffries usually called father of DNA fingerprinting. Next, satellite DNA shows high degree of polymorphism and forms the basis for DNA fingerprinting. So what is satellite DNA? Highly repetitive DNA is called satellite DNA. So in the genome, short stretch of DNA is repeated many times. That is called satellite DNA. Satellite DNA does not code for any proteins. It does not code for any proteins. So satellite DNA shows high degree of polymorphism. Polymorphism is nothing but variations in the DNA. So satellite DNA shows high degree of polymorphism and forms the basis for DNA fingerprinting. So satellite DNA is of mainly two types, mini satellite and microsatellite. So we already discussed this mini satellites and microsatellites in the last video. So it works mainly on the principle of VNTR. It's very important. So what is VNTR? Variable number of tandem repeat. VNTR is nothing but variable number of tandem repeat. So DNA fingerprinting mainly works on the principle of variable number of tandem repeats. What is variable number of tandem repeats? Before that, first you should understand what is tandem repeat. Tandem repeats occur in DNA where a pattern of one or more nucleotides are repeated and these repetitions are directly adjacent to each other. So tandem repeat is nothing but short stretch of nucleotides that is repeated again and again, and they are adjacent to each other. They are side by side. Then they are called tandem repeats. So here the tandem refers one after the other. So one after the other. Short stretch of DNA that is repeated many times and one after the other. That is called tandem. So tandem repeats occur in DNA where the pattern of one or more nucleotides repeated. So here if we observe this sequence, TA, TTG is repeated, and these repetitions are directly adjacent to each other, side by side. In between these two sequences, there is no another sequence. That's why it is called tandem repeat. Tandem means adjacent to each other, side by side. So one after the other. That is called tandem. So here we already discussed STRs and VNTR in the last video. Short tandem repeats and variable number of tandem repeats. Okay. Short tandem repeats comes under microsatellite. VNTR, variable number of tandem repeats, comes under mini satellite. So tandem repeat is nothing but short stretch of DNA that is repeated again and again and they are adjacent to each other is called tandem repeat. Short tandem repeat means if that tandem repeat is short, if that DNA sequence is short, then it is called short tandem repeat. What is variable number of tandem repeat? Variable number of tandem repeat is nothing but if the tandem repeats are variable in number and in population, then it is called variable number of tandem repeat. We will see here. Here, in case of short tandem repeat, which comes under microsatellite, so the number of base pairs will be 1 to 10. Here I have taken only 5 bases, 1, 2, 3, 4, 5 bases. And how many times the tandem repeat is repeated here? That particular DNA sequence is repeated how many times? 1, 2, 3, 4. 4 times here it is repeated. In case of this STR. Just I have given example. Now coming to VNTR, variable number of tandem repeat. The tandem repeats are different from one person to the other person, that is in the population, and different types of tandem repeats are present. Then it is called variable number of tandem repeats. So CAGC, GCA, TGG. This is one repeat. If you take another one, the same. TA, GC, CCG, A, GG, C. This is another. If that sequence is repeated again and again, and then it is called VNTR, variable number of tandem repeat. It differs from person to person. So variable number of tandem repeat is nothing but if tandem repeats are variable in number, then it is called variable number of tandem repeat. Here the number of bases will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Minimum 10. Maximum 100. We already discussed. In case of mini satellite, the number of base pairs will be repeated and the number of base pairs will be 10 to 100 and it may be repeated many times. And in case of microsatellite, STR comes under microsatellite. Here it is rich in AT. That's why you can, if you observe this, AT is TATTG. More A and T will be there. So AT, it is rich in AT. So in case of VNTR, it is rich in GC. That's why you can find more GC here. That you should keep in mind. Okay. So the VNTRs usually differ from one person to the other person. So by chance, VNTRs may be same in the individuals, but can vary in number of repeats from one individual to the other individual. If you take person 1, if you see here CA, GC, GCA, TGG, that is repeated how many times here? 1, 2, 3, 4, 5 times it is repeated. Right? So the number of repeats is 5. In case of person 1, the same sequence may be present very rarely, but it may be repeated 10 times. In second person, it may be repeated 100 times in the third person. It may be repeated maybe 200 times in the next person, or it may be repeated only 2 times in the next person, like that. So the tandem repeats are variable in number, and in population is called variable number of tandem repeats, and the number varies from one person to the other person. Though if by chance the VNTRs may be same in the individuals, it cannot be same in the individuals definitely, but in some cases it may be same, but the number may be varies from one individual to the other individual. That is, the same sequence, the CAG, CGC, ATGG, it is present maybe 10 times in one person, in another person 20 times, in another person 40 times, like that. And the copy number varies from chromosome to chromosome in an individual. See, the same tandem repeat CA, GC, GC, AT, GG may be present in the another chromosome. So there are total how many chromosomes are there? Total 46 chromosomes are there. So maybe chromosome 11 has this sequence and chromosome 22 also having the same sequence. But in chromosome 11, this tandem repeat, variable number of tandem, that is repeated how many times? 20 times. In chromosome, another chromosome, it is repeated maybe 100 times. We don't know. So it varies from person to the person. It also, the copy number also varies from chromosome to chromosome in the same individual also. Next, the number of repeats show high degree of polymorphism. So here the number of repeats which shows high degree of polymorphism. The size of VNTR varies from 0.1 to 20 kilobase pairs. Here variable number of tandem repeats, it shows high degree of polymorphism, which means variations. So the same sequence may not be repeated in the one person to the other person at the same position. So if you take the, for example, if you take this is the chromosome, this particular VNTR is present at this position of one person. The second person, if you take the same repeat, maybe you can find, but not in the same chromosome at the same position. It may vary. So definitely the number of repeats show high degree of polymorphism, that is variations. There is a meaning of this sentence. Next, the size of the VNTR varies from 0.1 to 20 kilobase pairs. So 1 kilobase pair contains 1000 bases. 1 kilobase pair contains 1000 base pairs. So 0.1 contains 100 base pairs. So 100 to 20,000 base pairs. 100 to 20,000 base pairs. So the size of the VNTR varies from 100 to 20,000 base pairs. Next, the child receives 50% of DNA from its father and the other 50% from the mother. So the number of VNTRs at a particular area of the DNA of the child will be different. So what does it mean? You know that here the child is formed by the fusion of the male and female gametes. Here definitely if you take the cell, if you are going to take the chromosome 7th chromosome. So you know that 2 sets of chromosomes are present. All the humans are diploid. One set is coming from the father, another set is coming from the mother. So you are going to identify the chromosome 11. So this is the chromosome 11. So 2 sets will be there. Chromosome 11 from father chromosome 11 from father. Next, chromosome 11 from mother. So these are the two chromosomes, one chromosome from father, one chromosome from the mother. So whether V and TRs are same in both cases? Definitely no, because this chromosome is coming from the father, this chromosome is coming from the mother. So at the specific area, here the V and TR will be like this. Here the V and TR will be different. The V and TRs differs from one chromosome to the other chromosome, and if you observe the chromosome 11 of the particular chain, so two sets are there, one from the father, one from the mother. If you see, so the V and TRs are usually differ. That only, see here, child receives 50% of DNA from its father and 50% from the mother. So, for example, if you are concentrating on the chromosome 11, one chromosome from father, one chromosome from mother. So, the number of V and TRs at a particular area, at this particular area of the chromosome will be different. So, this is about V and TRs. Now coming to steps involved in the DNA fingerprinting. Why we have discussed all these things? Because if you cannot understand the V and TR, it is highly impossible to understand the DNA fingerprinting. So this is about V and TRs. V and TRs is nothing but variable number of tandem repeats. So particular sequence which is repeated many times and differ from person to the person, from chromosome to the chromosome, and the number of times it is repeated also differ. It may be repeated 20 times in one person or 40 times in another person, maybe 120 times in another person, like that it differs. So this is about V and TRs. Next, coming to the steps involved in the DNA fingerprinting. Now coming to the steps involved in the DNA fingerprinting. How the DNA fingerprinting can be done. There are different steps how to perform the DNA fingerprinting. The first one is isolation of DNA. That is, DNA should be separated from the cells. Next, digestion of DNA by restriction enzymes. Restriction endonucleases are the special type of restriction enzymes which can cut the DNA at specific positions. These restriction enzymes that we will discuss in detail in biotechnology. So, digestion of DNA, that is breakdown of DNA by using restriction enzymes. Next, separation of DNA by gel electrophoresis. The DNA fragments can be separated by using the gel electrophoresis technique. Next, transferring of separated DNA fragments from the gel to NC membrane, nitrocellulose membrane or nylon membrane. This process is called blotting, southern blotting. Next, hybridization using labeled VNTR probes. Probe is nothing but short stretch of nucleotides with known base pairs is called probe. So, hybridization using labeled VNTR probes. Next, detection of hybridized DNA fragments by autoradiography. So, the DNA fragments can be detected by using autoradiography. So, these are the steps involved in the DNA fingerprinting. Now, we are going to study each and every one in detail. Isolation of DNA. First, you can isolate the DNA from the particular cell. So, this is the cell which is having the DNA. So, isolation of DNA. Next, from the cell, the DNA is to be isolated. Next, the DNA is subjected to restriction enzymes. When restriction enzymes are used, what happens? The DNA cut into number of pieces like this. Next, separation of DNA by gel electrophoresis. You cannot see the DNA with your naked eye. So, once you can cut the DNA by using restriction enzymes, you can subject those fragments to the gel electrophoresis. Next, transferring of separated DNA fragments. Here, the DNA fragments are separated. So, the separated DNA fragments can be transferred from the gel to the NC membrane. NC membrane is nothing but nitrocellulose membrane or nylon membrane. The fragments can be separated to the membrane like this. Next, hybridization using labeled VNTR probes. Here VNT probe is nothing but short stretch of known base pairs. So, here the fragments are there, but you don't know the number of base pairs present in the fragment here, and what is the type of the bases present here. So, you are adding the VNTR probes, known base pairs. So for example, here the ATGC is there. For example, you are adding TTGC, ATGC, TACG, like that, number of probes will be added. So the sequence is here ATGC. Among these probes, which probe can bind to the ATGC? Complementary TACG. TACG can bind and it can form the band like that. Okay, known probes are nothing but short stretch of DNA with known base pairs can be added. So once probes can be added, the specific probe can bind to the specific DNA fragment and can form the band. Next, detection of hybridization takes place. Hybridization is nothing but here one fragment can bind with the another complementary base pairs. Detection of hybridized DNA fragments by autoradiography. By using autoradiography, you can see the hybridized fragments. So these are the steps involved in the DNA fingerprinting. Now we are going to see the pictorial representation of the DNA fingerprinting. Now, for example, if some crime happened at one particular place, so we have to detect the criminal. We can detect the criminal by using the DNA fingerprinting. The main application of the DNA fingerprinting is forensic application. So you can easily detect the criminals by using the DNA fingerprinting technique. How? At one particular place, any crime has happened. One crime has happened. Now what they will... That particular scientist, he will collect the DNA. So the DNA can be collected from that particular place. That is either blood stains or hair follicles or sperm, whatever it may be. So the particular crime place, they will collect the cells or they will collect the DNA molecule. Once they can collect the DNA molecule, what they will do? They collect the cells or DNA. Next, that DNA is subjected to all these processes. Okay. Once all these 6 steps can be done, you can get these bands like this. Now, here, if you keenly observe, here this is the DNA isolated from the suspected person A. One person. We don't know, 2 persons are there, we don't know who did the crime. Okay. So this is the person A, from him DNA can be collected. This is the person B, DNA can be collected. This is the DNA collected from the place where crime happened. That is the sample. Some hair follicle or blood stain that you can find at the place where the crime has happened. You can collect the DNA from that particular place. So this is the DNA, right? So you know that how many chromosomes are present in case of humans? 46 chromosomes are there, right? 44 are autosomes, 2 are the sex chromosomes. Okay, total 46 chromosomes are there. Here they have mainly concentrated on the 3 chromosomes. So, chromosome 7, chromosome 2, chromosome 16. Okay, they have concentrated. No need to do the DNA fingerprinting for all the chromosomes. Okay, so you can take any 2 or 3 chromosomes enough to identify the criminal. So they have taken the 3 chromosomes from the DNA. I mean the DNA that is collected from the place where the crime has happened. Okay, this is the chromosome 7, this is the chromosome 2, this is the chromosome 16. So that is the 3 chromosomes they have isolated from the sample, that is a blood sample or something where the crime has happened. So this is the thing. So chromosome 7, chromosome 2, chromosome 16. You may ask why two chromosomes are there here. See, 1 chromosome from the father, 1 chromosome from the mother. So this is the paternal, this is the maternal. Paternal means chromosome from father, M means chromosome from mother. If you take the cell consists of 2 sets of chromosomes deployed, right? So 1 set from father, 1 set from the mother. So all the 44 chromosomes from all the 23 chromosomes from father, all the 23 chromosomes from mother, 22 chromosomes, autosomes from father, 1 chromosome, 1 sex chromosome from father, and 22 autosomes from mother, and 1 sex chromosome from mother. So like that they have isolated the chromosomes. Now if you observe here you are concentrating on the short tandem repeats. That is, we already discussed tandem repeats. So this is the first we will discuss about the DNA that is isolated from the crime sample. Okay, this is the DNA isolated from the sample where the crime has happened. Now chromosome 7, chromosome 2, chromosome 16. Chromosome 7 contains, so this is the paternal chromosome, this is the maternal chromosome. Paternal chromosomes contains 11 tandem repeats, maternal chromosome contains 9 tandem repeats. As I already said, all the individual does not have the same tandem repeats. You may ask, both are 7th chromosome only. Why different tandem repeats? Because one chromosome, this is also 11th chromosome, this is also 7th chromosome, this is also 7th chromosome. Both are 7th chromosome only. But this is the chromosome from father, this is the chromosome from mother of the particular cell. Okay, that's why tandem repeats also different. Here 11 tandem repeats are there, here 9 tandem repeats are there in case of chromosome 7. In case of chromosome 2, paternal chromosome contains 8 tandem repeats, maternal chromosome contains 7 tandem repeats in case of chromosome 2. Now coming to chromosome 16, this is the paternal chromosome contains 4 tandem repeats, maternal chromosome contains 2 tandem repeats. So this is the tandem repeat that we got from the sample. Now we can suspect the two persons, individual A and individual B. Person A, person B. We have to know who is the criminal. So what we have to do, again we have to isolate the DNA from individual A, isolate the DNA from individual B. Once you can isolate the DNA, you should follow all the 6 steps. Now we have to compare the number of tandem repeats present in the different chromosomes of the DNA isolated from the crime scene C and the number of tandem repeats present in the different chromosomes of the 2 different individuals, individual A, individual B. So you know that number of tandem repeats in chromosome 7, chromosome 2, chromosome 16 from the DNA of the crime scene C. Okay, now you can isolate the DNA from individual A, isolate the DNA from individual B, and you can compare the tandem repeats, number of tandem repeats with A, B and C. You can compare all the 3. So the tandem repeats that match, the tandem repeats of the particular individual can match with the crime scene C, then you can easily identify the criminal. Now you can see here number of, this is the individual A, this is the individual B, this is the DNA isolated from the place where crime has happened. Now chromosome 7, chromosome 7 contains 3 tandem repeats, chromosome 7 contains 3 tandem repeats in case of paternal chromosome. In case of maternal chromosome, number of tandem repeats are 4, 1, 2, 3, 4. Right. So here 11, 9. So chromosome 7 of the crime scene C cannot be matched with the chromosome 7 of the individual A. Right. Now coming to the here individual B. Here number of tandem repeats are 11, 9. Here also 11, 9. So here B is matched with C. Okay, this one. Next you can take chromosome 2. Chromosome 2 of the individual A contains, paternal chromosome contains 8 tandem repeats, maternal contains 7. Here also 8, 7. Here also 8, 7. Okay, in this case, chromosome 2, the VNTRs present in the chromosome 2 are same in all the 3 cases. That you should keep in mind. Next, coming to the chromosome 16. Here the chromosome 16 of the crime scene C, DNA from the crime scene C has 4 tandem repeats and at the maternal chromosome, and number of tandem repeats in the maternal chromosome will be 2 and paternal chromosomes will be 4. Here the number of tandem repeats 4, 10. 4, 10. Here 4, 2. Here 4, 2. So here also the tandem repeats present in the individual of the chromosome 16 cannot be matched with the tandem repeats present in the chromosome 16 of DNA from crime scene C. Why? Here paternal chromosome contains 4. Here also paternal chromosome contains 4, but maternal chromosomes contains 10 here, but here 2. It is not matched. Now we will see here the number of tandem repeats present in the chromosome 16 of the individual B of the paternal chromosome has 4, maternal chromosome has 2. Here same match. So if you keenly observe that number of tandem repeats or tandem repeats present in all the chromosomes of the individual B matched with all the chromosomes of the DNA isolated from the crime scene. See. So you can easily identify that individual B is the criminal. Here the bands. If you see the bands here number of tandem repeats. Here first you can take here 3 tandem repeats and 4 tandem repeats. In case of individual A 3 tandem repeats. Here yellow color, and here 2 tandem repeats. So likewise, if you see the bands of the person A, B and C, see here C can match it with the B in all the cases, but A cannot be matched with the C. So you can easily identify that B is the criminal, not A. So with that we can complete the DNA fingerprinting. This is how we can easily identify the criminals. Now coming to what are the applications of the DNA fingerprinting. There are different applications. First application is the identification of the criminals and rapists. Next, parent dispute solving. Next, determining the genetic diversity. So DNA fingerprinting is the very sophisticated technique which is used in the forensic applications. So this is about DNA fingerprinting. With that we completed the molecular biology. Thank you.